Good day, my name is Sherilyn Furman and I am here to discuss the genetic effects of selection, breeding methods for improvement of swine and reproductive and growth traits in swine. So let me first discuss the genetic effects of breeding. First is double muzzling gene. For example, in many species such as cattle, sheep, pigs, horses, and dogs and humans, the myostatin gene is known. This locus has a recessive allele in homozygous animals gives rise to the double muzzled pattern. A breed where the fixation of this allele is aimed at is Belgian white blue cattle breed. The breed is famous for the heavy carcasses, the thick muscles, and the high percentage of meat in the carcass. However, a high percentage of the females that are homozygous for the double muzzle allele of the myostatin gene cannot give birth in a natural way. Those calves are born through cesarean section, leading to a severe ethical discussion in many countries. In other species, the animals homozygous for the myostatin gene give smaller problems when they give birth, and cesarean sections are required. Second is the milk protein genes. Another example of a gene with a known major effect occurs in dairy cattle. In dairy cattle, a number of milk protein genes are known from which the alleles have a different effect on cheese yield. For example, a beta-lactoglobulin alleles gene located at chromosome number 11 have a marked effect on the efficiency of cheese production. Cows with the BB allele are the favorite ones for cheesemakers. The DG81 alleles gene located at chromosome number 14 influence fat percentage in milk and the milk fat composition. The K allele increases fat and protein percentage and fat yield, while it decreases milk and protein yield. And very importantly, the milk composition from cows with K alleles is different. They produce more fatty acids that are considered to be less favorable for human health. The next is meat quality gene. In pigs, that halothane gene, located at chromosome 6, is known to influence the stress susceptibility and meat quality. Research is focused on chromosome 6 in pigs where genes are located that influence the androstenone production in boars. Androstenone is responsible for an awful boar taint in the meat of intact boars. Up to now, boars are castrated to avoid this boar taint, but selecting against boar taint is a better approach for the welfare of the boars. Next is fertility genes. In sheep, several genes are described that have an influence on the litter size. An example is that of the Borula gene present in Australian Merino has a marked effect. The heterozygous carrier produces 1 lamb more and the homozygous carrier 2 lambs more per liter. This allele is now also present in the Dutch Texel breed by crossbreeding Merino carrier rams of the allele with Texel ilk and backcrossing the crossbreeds and Texel sheep. Next is color genes. In all species, a lot of attention is paid to the inheritance of the color of the coat. The color of the coat is an important trait in the recognition of breeds. Breed association often have strict rules for the required color pattern. In companion animals and animals used for leisure purposes, breeders pay a lot of attention to the inheritance of color in breeds for special phenotypes. In the past, many genes and alleles are described that are involved in the color inheritance. First, we start to describe a few genes that play a role in coat color in ruminants, and then we mention per species additional particulars. Next, let me discuss the effects of selective breeding. Selective breeding is choosing of parents with particular characteristics to breed together and produce offspring with more desirable characteristics. So the effects of selective breeding are the following. Some of the offspring may even be taller than both parents because they inherit a combination of different tall gene variants from each parent, and together these make the offspring taller. With repeated selective breeding over multiple generations, this population will get taller and taller. Selective breeding often results in a population of animals or plants with very similar genetics. Similar genetics means that a population will have the same strengths but also the same weaknesses. And last is infectious diseases are more likely to spread through the genetically similar populations because they are vulnerable to the same disease. Now let me discuss the breeding methods for improvement of swine. This guide from the FAO provides small-scale or backyard farmers information on how best to breed your pigs. First is pure breeding. Pure breeding is mating purebred individuals of the same breed. The progeny has the same genetic makeup. The major objective of pure breeding is to identify and propagate superior genes for use in commercial production, primarily in crossbreeding programs, as well as to propagate and identify superior females for maintaining valuable genetic material. Furthermore, crossbreeding will not be worthwhile unless superior purebred individuals are used. Next is outbreeding. Outbreeding is mating individuals of the same breed but who are less likely related than the average of the breed. There should not be a common ancestor for at least 4 generations back in the pedigree of the boar and the females with which he is mated. It is a useful mating system in purebred individuals. Next is inbreeding. Mating between individuals of the same breed which are more closely related than the average of the breed. This could be between as close individuals as full sibs or sire, daughter, mother, son. Pure breeding is a special kind of inbreeding. The effect of inbreeding is the concentration of common genes in the offspring. This high frequency of homozygous gene pairs applies to both desirable and undesirable traits. Many undesirable traits, such as hernia and cryptorchidism, involve recessive genes. Thus, inbreeding perpetuates their expression phenotypically. Inbreeding causes decrease in litter size and increased mortality. Inbred sows are inferior in milking and mothering ability. It delays sexual maturity in gilts and boars. Inbred boars have less sexual libido. Inbred gilts have fewer eggs during oestrus and farrow smaller litters than those outbred. The effects of inbreeding are the following. So with newly born litters about 12 piglets, male and female piglets taken by another farmer to his home, breeding between the same siblings, female giving birth about 9 piglets, about 25% number of smaller and weak piglets are born as compared with first sow, and some of them were born dead. Male and female piglet taken by another farmer to his home, breeding between the same siblings, about 6 piglets. About 50% number of very smaller and weak piglets born as compared with the first sow. Some of them were born dead. Crossbreeding, mating 2 individuals from different breeds, thus introducing into the progeny a gene combination that is different from that existing in either parent or in the breed of either parent. Crossbreeding can involve 2 or more breeds, depending on the desired result. The sole purpose of crossbreeding is to take advantage of the observed improvement in performance of the progeny about that of either parent hybrid vigor or heterosis. The effect of crossbreeding are the following. First is local sow with her piglets, exotic sow with her piglets, female piglet from local sow and male piglet from exotic sow are taken by a farmer to his home for breeding purpose. Breeding between this male and female pig. Healthy and bigger body size piglets are born from the local sow. All male piglets are sold and few selected crossbreed female are kept for breeding. Another exotic male which is not in relation with the herd introduced for breeding. Breeding between them. Very healthy, strong, active, and fast-growing piglets are born. Now let me discuss the reproductive and growth traits in swine. Measuring reproductive performance. The reproductive herd is for the multiplication of maternal and paternal lines that are prized for economically valuable reproductive traits, or traits that are generate desirable offspring reliably. Typically, prized productive traits that include litter size, particularly total number of pigs born and pigs weaned, weight of the litter of piglets at birth and weaning, wean to estrus interval or WEI, and pigs per sow per year or PSY. In more recent years, length of time a sow remains productive in the herd, longevity or survivability is increasing in priority for major genetic lines. These traits are prioritized due to the impact on value of the sow to the herd. Commonly, these records are collected to measure productivity in the reproductive herd can be defined as follows. First is litter size. The number of pigs per litter. As the number of pigs born per litter increase, generally, there is an increase in the number of pigs born alive and the number of pigs weaned. This number is currently scrutinized as a primary economical focus, as in recent years, there has been evidence of reduced birth weights due to increased litter sizes, which is associated with increased mortality rates and decreased growth rates in offspring. However, in the same period of time, offsprings have been selected and now grow at the fastest rate in history. Mortality rates have generally remained the same. Litter size remains a top priority for most pig producers. Next is total born alive. Total born is the number of pigs a sow has, including born alive, stillborn, and mummies. The number born alive is all piglets born alive, including number of pigs born still or stillborn fully developed but not alive at birth, and mummies are partially decomposed due to death early in gestation and before full development. Total weaned. The total number of pigs an individual sow weans. Marker of sow's ability to provide enough milk and mothering ability to rear pigs. Birth and wean weight. Birth and wean weight or birth weight is the weight of piglets born alive within the first 24 hours. Typically, this includes all piglets that were born alive, even if some of those pigs died before weighing. Wean weight is the weight of the piglets that were weaned from 1 litter. Weights can be individual but are usually collected by producers as an entire litter. Next is the wean to estrus interval or WEI. This is the amount of time that passes from the time a litter is weaned from a sow to the time she returns to estrus. A good sow should be reliable in her ability to return to a productive heat of estrus in a timely manner post-weaning, usually 4 to 6 days. It is very common producers to give sows up to 7 days post-weaning to show signs of estrus. After 7 days, the opportunity to catch a reliable heat is usually dependent on the sow's ability to enter back into normal estrus cycle. Depending on your system, You may wait until the next cycle, 18 to 21 days, until watching for heat and trying to breed again. The next is the heat no service. As explained in the name, a heat or signal of readiness to breed is observed in the sow. However, she is not inseminated, breed or serviced by a boar or artificial insemination. Instead, she is allowed to continue in the estrous cycle, 18 to 21 days, until her next heat of estrus period is reached. Purposely skip heat is different than a missed heat. Purposely skip heat stages are associated with management decisions that are for the benefit of the sow or the system. Missed heat is either lack of management or a reproductively unpredictable sow. In either case here, a missed heat generally contributes to a poor understanding of herd performance. Next is days non-productive or DNP. The number of days that a sow is not in gestation or pregnant, lactation, or the 7-day window post-weaning. This number can be used to evaluate overhead cost of caring for sows in the herd and act as a measure of reproductive performance when culling decisions are made or management changes occur. Next is pigs per sow or per year, also called as PSY. It is the number of piglets born per sow in a given period, expressed on a yearly basis, divided by the average herd size at mating. This is often used to broadly evaluate sow productivity within the entire herd and across parties. PSY has traditionally been the benchmark of reproductive efficiency. However, current big production models place less emphasis on this single measurement. Next is longevity. How long a sow stays productive or continues to have litters in the herd. Longevity of sows in pig operations can and should be measured across a variety of indicators of sow removal, such as replacement rate, culling rate, and percent of gilt in the herd, and mean party, number of litters of females in the herd. Each maker of removal can indicate a different reason for sow existing the herd and should be carefully considered. So these are the resources of my report, and thank you for listening. I hope you learned something.